Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Lucas Klein and Dirk Mueller from Sartorius.
Yes, hello. Thank you for this nice introduction, Leah, and welcome everybody to our webinar today. What we would like to introduce you today is tools for cell line development, how we can accelerate process optimization by combining EMBER 15 cell culture with octet titer measurements as a means to improve your cell line development workflows with analytical tools. So let's have a look what we're up to today on the agenda. First, we want to introduce you to our EMBER 15 system setup and how you can use it. Then we want to lose a few words on the octet technology for biomolecule quantification and how we can apply it in our bioprocess environment. Then we take a break for a short survey where we would like to get some feedback from you before we head on to the application examples of this setup with media screening and process characterization before we finish off with a summary and move to the Q&A session. So, and with that, over to Lucas, please, who will tell you something about the EMBER 15 system.
Yeah, also from me, a very warm welcome to this presentation here today, and thank you very much for the nice introduction. So let's start right away, and let me introduce you briefly the EMBER 15 system. With the EMBER 15 system, you are able to perform automated multiple bioreactor cultivations in a 10 to 15 milliliter scale. Depending on your system, up to 48 cultures can be controlled. The whole system consists of a working station which is installed in a biological safety cabinet, single-use bioreactor, and a laptop computer, including the EMBER 15 software. With this software, you are able to control the whole device, set up your cultivation protocols, and much more. So a key automation feature of the EMBER 15 is the liquid handling robot. With this robot, samples for online and offline analytics can be taken fully automatically, such analytics can be pH measurement, so for instance with an integrated analysis module, metabolite, product concentration, and cell count and viability. Due to the EMBER 15 exclusive bioreactors, which bear a DO and pH spot on their bottom side, on-demand DO and pH control can be realized by an integrated optical spot reader individually for each bioreactor. And the software evaluates then the signals and reacts accordingly to maintain giving DO and pH set points. The EMBER 15 culture stations, to which each culture station holds up to 12 bioreactors, also allows the user to control temperature and steering to maintain the cells happy and healthy. Yeah, compared to cultivation approaches like shake flasks, the EMBER 15 is more representative to complex systems like bioreactors due to the capability of easy process control like DO and pH and steering. It may also be used to predict process parameters and behavior in large scales like benchtop format. Applications of the EMBER 15 could be clone selection, so for instance in a CLD process, media and feed screening in media development, and early process optimization. Several processes like batch, fed batch, and even perfusion mimic can be conducted with the device, either with mammalian or insect cells. The systems can be used for cultivation of suspension cell lines or adherent cell lines, which can be attached to microcarriers. And now that I showed you the very basics of the EMBER 15 system, I'd like to introduce you to the octet technology. As I mentioned before, the octet is able to use multiple sensors at one time, which makes it No, sorry, the Octet is a device for multiple kinds of analytics, and its principle is based on the biolayer interferometry. The system uses specific biosensors coated with a ligand like protein A to facilitate the immobilization of the analyte. So, for instance, an antibody, which we can also see here on the right. In one analysis round, so multiple samples can be measured at the same time when using multiple biosensors. Using the dip and read methodology, the real-time interaction monitoring can be realized using the According's Octet software. So, as I mentioned just before, the Octet is able to use multiple sensors at one time, which makes it a device carrying out measurements in a high throughput manner. As we can also see in relation to other techniques like ELISA or HPLC, the Octet technology is a cost and time saving methodology, as it is here depicted in the table. Moreover, the handling is very easy and sample preparation is kept at a minimum. So, for instance, no protein A purification step is needed for monoclonal antibody products. With the Octet technology, the operator can get many outcomes from one measurement, as the device can determine A, binding kinetics, so what is the rate of interaction of ligand and analyte, B, the binding affinity, so how strong do the molecules bind to the ligand, the binding specificity, which tells me do the molecules interact with the ligand in the first place, and D, the concentration, so how much analyte is in the sample. What we see here is a conducted Octet measurement and the related binding kinetics. This curve actually is displayed in the Octet software at real time during the measurement. So even during the measurement, the operator gets a hint which sample contains more product or analyte. And this is also indicated here by the arrow. So we see if the slope of one curve is higher and the saturation is reached faster, the operator can then conclude that there's more analyte in the sample. And for further evaluation and assessment of an instant product concentration, a standard curve, which we can also see here, has to be registered in the software once for every product. So once the operator has registered a standard curve for one product, one can use that for every measurement in future if it's the same product. And now my colleague Dirk will shed some light on the Ember Octet integration.
Yes, thank you very much, Lucas, for introducing both the Ember and the Octet. So the important question is, of course, now if you have a real bioprocess, how can we bring the two systems together? How do they interact? Just an example, you see on the left-hand side here this Ember 15 laptop that is used to program the Ember 15 system. And for example, you can now program that every day, every bioreactor would be sampled, and you can use that sample in some pre-processing, usually spinning down the cells, and then without much ado and after some dilution, do this measurement in the Octet, for example, for product titer determination. And the key advantage is really that for a 48 well system or 48 bioreactor system, as we have it for an Ember 15, you can actually do this now in almost only about half an hour the whole time, which is a real improvement over many other methods which are available on the market. Okay, so much for that. At the moment, I think we've told you a little bit about how the two systems operate. And before we now go into our application examples, we would like to ask you a few questions in our survey. Thanks go for you, Leah.
Okay, great. So your survey question should be on the screen. You can use your mouse to go ahead and enter your answers. The first question is, what is your main work area? Process development, cell line development, media development, or other? And do you use parallel mini bioreactor systems? Already using, would love to get started, or not planned? And are you employing a method for product titer determination? protein A, HPLC, octet system, or ELISA, or other? And finally, what are your main pain points in titer determination? Low throughput, suboptimal accuracy, unspecific binding, cumbersome sample preparation, or other. So if you want to go ahead and enter your answers for the other in our Q&A box, that would be helpful. But go ahead and take the time to answer those questions on your screen. And then, like I said, if you've answered other, you can go ahead and enter your answers. You can just enter them with a semicolon between them if there's multiple others, and we'll figure out which one it goes with, I'm sure. So thank you very much, and we're just going to give you a couple more seconds to go ahead and answer those. And send it back to Dirk.
Okay, yeah, thank you, Leah. Now let's continue with some of the application examples here. Oops, I think we're over. And the first application is on media screening. Lucas, the floor is yours.
Yeah, thank you very much, Dirk. So the first application example I want to show is a media screening experiment. Our challenge here was to find a process how to quickly elucidate the one media which provides the capability for a given clone to reach the highest titer. The other part was that we had to compare different media in respect to product concentration. So we questioned ourselves, so how are we able to get reliable results, like multiple time points quickly? And how do we do this without putting much effort in it? So why is this relevant? Yeah, it is relevant because as media or cell line developers, we need to compare media from different vendors to reach the best product concentration. And B, we need to compare different in-house media variants during our media development. And those aspects and workflows are very time consuming and a lot of work. So basically, our solution was to combine a device which is capable to cultivate multiple reactors at the same time with automated sampling, so the Ember 15, and a device which is able to determine product concentration in a high throughput manner, the octet. For this, we set up an Ember 15 experiment with 2 clones and 5 media. We collected the samples automatically every day and analyzed them all at once for product concentration using the octet device. So the results for clone 1 are shown here. As we can see, similar VCCs were reached during the process, but with media 2, the lowest viability was observed. Also, media 3 showed a weak viability at the end of the process. On the upper right part of the picture, product concentration is depicted, and these data reveal that using media 5 results in the highest product yield of clone 1. And looking on the lower right graph, it is shown that the specific productivity could be maintained quite stable for nearly all of the tested media. So when we have a look on the results we got for clone 2, it is evident that highest cell counts could be achieved with media 1 and 2, and the data also reveal that viability drops earlier for clones 3, 4, and 5, suggesting that this clone seems to feel more comfortable in media 1 and 2. And talking about product concentration, there is an obvious divergence from day 8 on with media 2 giving the highest yield. So, but all in all, it seems like the best media differs between clone 1 and 2, since we saw that for clone 1, media 5 was the way to go, and here we would have media 2. Well, I'm sorry. What we can say is also that it is clearly a benefit to couple Ember 15 and octet technologies in a media screening experiments, since we were able to analyze quickly process dynamics of specific productivity for hints to media-induced limitations. And since we could observe a divergence in specific productivity with different media, this gave the first identification to a potential limitation in late-stage process, which again accounts for combining Ember 15 and octet technology. And just a quick note, I also want to point out that all these experiments were conducted with the Cellcar CHO DG44 clones, in-house clones. Coming to main benefits of the octet analysis when subsequently done after an EMBER-15 run, we can remark a significant reduction of hands-on time compared to HPLC with a decrease of 75 or 80%. Another point is that the analysis time can be reduced by more than 90% in relation to HPLC. And last but not least, there is a reduction of consumables and above all, there is much less buffer and no buffer preparation needed. And to further reduce costs, the biosensors can also be reused during a measurement of a 96-well sample plate, which leads to a sample amount of 72 samples per plate. And now I want to show you another application example, which is going into the direction of a process optimization. So here, the challenge was to identify the best EMBER-15 process settings for our CLD platform process with focus on specific productivity or also QP. Typically, an identification of best process parameters for a platform process would mean screening of several clones and conditions which then automatically lead to many experiments. So in the first step, we identify the most sensitive parameters who may affect QP. And in the second step, a reduced set of settings could be applied to multiple clones. As I mentioned before, such an approach leads to many experiments, which first of all have to be planned, since lots of conditions have to be tested. Then they have to be conducted and further analyzed. So to minimize the effort and save time and resources, a tool or workflow is needed to set up experiments quickly and efficiently. The workflow we identified to be suitable for our experiment is shown on this slide. So we actually used 3 different systems and combined them for a smooth and efficient experimental workflow. So first, we defined a working space with mini and maxima and center point values for the parameters we wanted to change. Then secondly, we used MODIS software for definition of our design of experiment. In total, we got 12 conditions which we ran in duplicates. This led to a total run number of 24. Then we inoculated an EMBER-15 experiment, and after the run, we fed the results of the EMBER-15 and octet, so like cell count, viability, product concentration, and QP, into the MODIS software to find out the best process settings respective to QP. Before we come to the result of the DOE, let me briefly explain our setup. So we identified steering speed, starting volume, and ballast gassing as critical parameters affecting scalability of QP and used a reduced combinatorial design. So this design, or this kind of design, let us having a good balance between the factors investigated with limited number of experiments and replicates. As depicted here in the table, the steering speeds from 1050 to 1650 RPM were used, as well as a range of a ballast gassing from 0.05 to 0.25 milliliters per minute. And additionally, different starting volumes from 12 to 40 milliliters were included in the DOE. And here, another quick note. So as for ballast gassing, we understand a constant base airflow rate, which ensures fast lowering of dissolved oxygen content early in the process and limiting the pCO2 buildup by CO2 stripping, so which is especially relevant in late process phases. So just to remind you quickly on the workflow of the experiment, during cultivation, cell counts were assessed and afterwards titer values were determined. Specific productivity was then calculated and all determined response data was then transferred into a MODI program for DOE evaluation. The outcome of this evaluation should then reveal the best process settings for our CLD platform process with respect to QP. Before moving to results, I have to point out again that these parameters are shown for one Cellcar CHO DG44 clone here. Okay, coming to the DOE evaluation. So this contour plot shows how specific productivity is impacted by ballast gassing and steering speed. Here we look at those parameters in combination. And using MODE, a partial least square model was fitted into the data. This kind of model is used for constructing predictive models when the factors are many and highly collinear. So for this clone, it seems that the QP is important at low steering speeds and increased ballast gassing. Anyway, the positive impact of high ballast gassing goes into the other direction when applying also high steering speeds. Those outcomes gave us the hint to operate at lower constant ballast gassing to maximize the specific productivity. Talking about high steering speeds and gassing, we also had to consider possible negative shear stress effect or a less efficient gas transfer caused by impeller flooding, which both may have an impact on our QP. So this would be depicted in the upper right corner in the contour plot, by the arrow. So when we look at the viability, the data reveal that there is a slight tendency of faster declining of viability at higher gassing and steering. And additionally, we didn't see any declining of the O at those high conditions. This gave us then the hint for a potential shear effect, but not for oxygen limitation due to impeller flooding at high gassing. So looking at on parameters individually can also give some insights to understand the model and how to put a factor of importance to them, to the assessed parameters. This main effect plots show how starting volume here on the left and steering on the right affect QP individually, positive or negatively. The starting volume found to have a negative effect on the QP the larger it gets, but only to a certain extent. For steering speed, an increase of this parameter seems to have a positive effect, but only up to a certain value, and then tends to lead to a decrease of QP. Also, it seems that this parameter is a more sensitive parameter as starting volume, since the impact range is higher here, as we have a range from 30 to 38 picograms per cell per day. And so from this data, one could draw that the best settings to maximize QP would be 12 milliliters starting volume and around 1,500 RPM. So in order to summarize the application example of process optimization, we could draw several benefits. Since we combined 3 efficient systems or tools, we were able to do fast process optimization with A, MODE, which gives us a fast COE definition and evaluation, B, the Ember 15, with which we have a controlled cultivation of multiple bioreactors in a small scale and automated sampling in the absence of an operator, and C, the Octet, which serves with an easy sample preparation and subsequent quick sample analysis. So as in process optimization, many experiments are required, as I mentioned before, and thus many samples have to be analyzed. Octet delivers a high throughput and operator-friendly solution by minimizing the effort in regard to determining tighter or specific productivity in the subsequent relation to the best process settings, which are described in this table. In this table, we can see that the parameters predicted by MODE are in a nice consensus with the data shown before and may lead to a good specific productivity for this clone. And now with this, I would like to hand over once more to my colleague Dirk to summarize this presentation. And thank you very much for your listening.
Yeah, thank you, Lucas, for the nice application examples. Let's have a look at what we have seen here today. So first of all, we have seen that combining the Ember 15 with the Octet analytics enables us to do a time-efficient execution and analysis of multi-parallel cultivations. The real advantage is here really that we can save a lot of in-person time, up to 75%, for an otherwise quite tedious time series analysis of multiple cultivations. Yeah, for example, when you would like to assess not only the final titer, but really specific productivity dynamics during a process. What are typical applications where we can use this combination? Well, first of all, typical scenarios are like media variant screening for a process improvement. If you're doing cell line development, selection of the proper production clone, when you want to know more than just the final titer, and for example, also process optimization using a DOE approach, where you are looking at different responses or look, for example, at cell-specific productivity. As Lucas has also mentioned, what is really nice is that the Octet titer analytics and the model DOE software really allow for an easy joint evaluation of processes, looking at really non-intuitive optima in the process conditions, even in a single Ember 15 run when you would like to optimize, for example, specific productivity. Yeah, and with that, I would like to hand over basically for your follow-up questions later on to the experts in our team. So Alison, if you have any questions regarding the Ember 15, Ivan is the right guy to talk to if it comes to the Octet systems, if you have further questions on that. And for application examples, please reach out to Lucas if you have more questions there. I would like not to close without acknowledging the people who supported us in compiling the data we've shown you today. So Eva and Connie were instrumental in collecting the data. Timo supported the data analytics, and John and David supported us, especially with the Octet part of this talk. With that, thanks a lot for your attention, and we're happy to move over to the Q&A.
Okay, great. So we've got a few questions here. So the first question is, does the Amber 15 serve as a good scale down model per your data for PC purpose?
So yeah, I should unmute first. So the, by PC, I assume this means process characterization. So I think in our hand, I would say it depends. So in most cases, we use the Ember 15 more for screening type applications. If you want to get a good idea and a first hint as to find out what the boundaries could be that you're looking at for your process parameters. If you want to go to larger scales, in particular, we recommend for a finer tuning to use the Ember 250 type systems, which has some advanced control features that make it even easier to translate it to larger scale cultivations.
And can you please comment on how Amber 15 and Octet can be used for lentiviral vector production?
Yeah, okay, that is a good one. I think we know we have some applications upcoming that are looking at AAV type applications. I have to admit, I'm not quite sure what is the status currently for the lentiviral applications. I recommend to reach out to Ivan Krylov, which is mentioned on the contacts page, for further details, but I can also do follow up internally and reach out to you after the webinar to get you over the answer to that.
Perfect. And is the Amber 15 process scalable to pilot manufacturing scale?
Yeah, I think this is pretty much a bit similar to the question we have had before for the process characterization. As said, I think in many cases we see that we get very reproducible results when using the Ember 250 for the upscale. That being said, we can, with some tuning of the Ember 15, also get quite good predictable results at the Ember 15 stage. However, there is some additional work needed for every clone that you need to scale up typically. So depending if you have a platform process or just a particular clone and how much time you invest, you can use the Ember 15 or rather go to the Ember 250 instead.
Okay. So I think we have time for just like two more questions. I know we got started a bit late. So the first question is, how many runs were actually performed for the process optimization designer experiments?
So there were actually 2 runs performed for the process optimization part. So and each duplicates, so which means that we had in 48 different let's say conditions.
Okay, So we have time for this last question. Go ahead and continue to type your questions in. We'll pass those to the speaker, and they can follow up with you directly. And if we didn't get to your question, we'll be passing those to the speakers as well. So what benefit does it bring to use the sensor regeneration capability?
So actually the sensor regeneration capability brings, it reduces actually, first of all, the costs. So we can, these sensor, you can like regenerate and use them like for 9 different times. So in a plate you have a row of 9 samples, and you can dip one sensor 9 times into one. So like you have analyzing regeneration, and then after that you can go into the next well, and this is actually the main benefit for it. So this is quite good.
Okay. Well, thank you, Lucas and Dirk. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.